Hi guys, today we are going to talk about the new lesson, which is biological molecules. These are the points which are needed to be covered according to the IGCSE syllabus. Biological molecules. Before starting to explain biological molecules, we need to know that the molecules around us divided into organic molecules and inorganic molecules. For the organic molecules means they contain carbon and hydrogen, but the inorganic molecules, these are the molecules which do not contain carbon atom. Our example is water and some mineral salts as NaCl or sodium chloride. If we go back to the organic molecules, they are large molecules and also they called biological molecules, which our lesson talking about. So this is the main point of our lesson. They may contain other elements other than the carbon and hydrogen. So the main elements are carbon and hydrogen, but we can add oxygen or nitrogen. So the examples of these biological molecules: carbohydrates, proteins, lipids, and nucleic acid. Firstly, the carbohydrates, they contain, because they are biological molecules or they are organic molecules, they contain carbon, hydrogen, and also they contain oxygen. These carbohydrates can be classified into monosaccharides and polysaccharides. By the way, we have other classification according to the number of sugars: monosaccharides, disaccharides, and polysaccharides. But according to your lesson, we have only two classification: mono and polysaccharides. Monosaccharides, they are the simplest sugars or the simplest carbohydrates. They have general formula CH2O over N, and this N the number of carbon atoms. So our example is glucose. So we have glucose C6, 6 carbon atoms. So here the hydrogen will be 12 because it's H2O, and oxygen the same number of carbon atoms. So this is our example is the glucose. The properties of monosaccharides: they are soluble in water. So because they are soluble in water, so they easily transported by the blood and dissolved in the cytoplasm of the cell. What is the function of the monosaccharides? The main source of energy, the main source of energy. And as I told you, our example is glucose. This is the shape of the glucose, the cyclic form of glucose. For the polysaccharides, they are formed of many monosaccharides combined together. So if I have different glucose or many atoms or many molecules of glucose, they combine together to form large molecules of other carbohydrates, which is complex carbohydrates or polycarbohydrates. And because they are complex, because they are polysaccharides, so they are insoluble in water. But what is the function? They are forming of some parts of the cell, like we have an example like cellulose, and as I told you before, cellulose is the main carbohydrate to form the cell wall of the plant cell. Also we have starch. This is starch which is stored inside the plant cell. This is the plant carbohydrates which is stored energy, because by the time if we need more energy and the cell consume up this glucose, so the starch is broken down by hydrolysis into glucose to give energy which is needed for the cells. Also we have other example which is glycogen. This is the complex carbohydrates or the polysaccharide which is stored inside the human cells, especially in the liver and muscle cell. So if we have one glucose, it will be mono because it's only one, but poly means many combined together and forming the polysaccharides. Secondly, we have the lipids. Lipids or fats. This is the common name of lipids. Each molecule of lipid is formed of 3 fatty acids and glycerol. So if I have 1 molecules of glycerol combined with 3 fatty acids, and we can divide these lipids into fats or oil. What is the difference? Both of them are lipids, but the fats are solid in room temperature, but the oil is liquid at room temperature. What are their function? Their main function of the lipid, they are source of energy, and by the way, they give energy more than the carbohydrate itself. But the carbohydrate is the fastest source of energy in our body. If we have carbohydrate and we have fat in our body, firstly we will start to get the energy from carbohydrates, and then from the fat if the carbohydrate used up. Also, they are used in myelin sheath in neuron. Neuron is the nerve cell, and nerve cell responsible for transmit of the neurotransmitter from part to another. And because of this, to prevent the shock, we need to cover this myelin or this cell with an isolator, which is called myelin sheath, which is formed of fat, and we'll explain this in details later in central nervous system. And finally, it's used for making up the cell membrane. If you remember, we described the cell membrane as a thin layer of fat and protein. So we have cell membrane which made up of fat. Proteins, this is the third one, formed of long chain of subunits. What are these subunits called? Amino acids. We have different amino acids, as you see, which are combining together to form long chain of amino acid, which is called protein. What is the function of protein? Protein used for growth, used for nails and hairs. Enzymes, if you remember in the enzymes lesson, we said they are protein in nature because the active site is formed of protein. Cell membrane, thin layer of fat and protein, so used for building up of cell membrane of the cells. Binding site of antibodies, and antibodies your immune system, which acts against the microorganisms. The last one is the nucleic acid. And nucleic acid, our example is the DNA, which is double helix. As you see, this is the double helix because it's twisted, but actually they are 2 strands, as you see, and we have bases or nucleotide which arranged in each one. These bases sequence form code which carries genetic information. So in DNA we have code, we have sequence of bases which is called code. This code is transported from one generation to the next generation. That's why you look like your father or your mother, because you carry some of these genes which transported from your parents. Here, as I told you, we have the bases. Actually they are called nucleotide. Each nucleotide is formed of base plus sugar, which mainly ribose, and ribose means just 5 carbon atoms and phosphate group. The nucleotides, they are different from one to another according to the bases, because actually we have 4 bases: A, which pairing up with T, and C, which pairing up with G. So for example, if I have here in one strand this sequence of bases or this sequence of nucleotide A, C, T, G, C and T, what is the pairing up of the other nucleotide in the other side? So A pairing with T, G, yes, A, C, G and A, and in between them we have cross-link. This cross-link mainly is hydrogen bond. It's hydrogen bond. Okay, so after pairing of the 2 strands, it twists to give this shape, the shape of DNA, the shape of DNA. Finally, we have the water. As we said, it's inorganic molecule, but it's important for the living organisms. Water considered as the biological solvent for the whole living organisms. Why? Because water, as I told you, is solvent for chemical reaction which transport dissolved substance into and out the cell. So we have some substance which needed to be inside the cell, how carried by the water, and then some toxic substances which can be or needed to go out the cell, which carried by the water. So dissolve them on the water and carry them out or in the cell. Helps in buffer the temperature. Why we need to buffer the temperature? As I told you, we are living organisms, so we have enzymes which are biological catalysts which controlled by the temperature. So the temperature should be adjusted to prevent any denature of the enzyme by increasing the temperature or slowing down the activity of enzyme to by decreasing the temperature. How can we buffer this? By the presence of water. By the presence of water. That's why it helps in digestion. It's very important because digestion controlled by different enzymes. So we need to buffer this. Dissolved mineral salts and the excess of mineral salts carried by the water in our urinary system and carried outside our body by excretion. That's why the water is our vital or important biological solvent for the whole living organisms. The last part we have is the practical work. How can we do the practical work for each molecule or each biochemical? We have different biochemical, and these are common in paper 6, which are important. If we start with the first one, the starch. Okay, so we have different points which needed to know. Name of the test: starch test. Reagent: iodine solution. It's our indicator for the starch. The test: get the solution and add few drops of iodine solution. What are the results? If we have starch, so it will be blue, black. If we don't have, so it will be orange, brown. Very easy. So how can we indicate if the starch found in the solution or not? By getting the solution, add few drops of iodine solution and then test it or test the color. Secondly, we have reducing sugar or glucose. What is the name of the test? Benedict test. Reagent: Benedict reagent or solution. How can we do this test? By getting equal amount of or equal volume of the food solution and the Benedict solution. So for example, if we have test tube and we add 2 cube centimeter of food solution plus 2 cube centimeter of Benedict solution, and by the way, Benedict solution is blue in color. What else? Heat them. Sometimes we add them in water bath and check the change of the color by heating. Check for heating for few minutes, around 2 minutes or 3 minutes, and check the change of the color. If we have negative, so the solution will be blue. Why? Because this is the color of the Benedict solution, but any change other than the blue, it will be positive. What are these? It will be maybe orange or brick red. So we have different color in between these two colors, between orange and brick red. Any color, generally any color between this one or any color other than the blue, it will be positive. So it means the glucose or the reducing sugar is found in the food solution. And by the way, the change of the color from orange to brick red according to the concentration of glucose. It means orange or yellow, it means the concentration of the solution is diluted glucose solution, but brick red, it means it's concentrated glucose solution. Third one, protein. So the protein by purity test. How? By the purity reagent or solution. We'll add 2 cube centimeter of food solution plus 2 cube centimeter of NaOH. Shake well and then add few drops of 1% copper sulfate, which is blue in color. So what are the results? If we have negative, it means no protein, though the color will be blue, the color of copper sulfate. But if we have positive, the protein is present, so the color will be violet or purple. And it's very clear, by the way, we did this in the lab in our school. The next one is fat. What is the name of the test? Emulsion test. What is the reagent? Ethanol. Fat cannot be dissolved at all in water, so we have the fat solvent, which is called ethanol. So in test tube we'll add the food solution and ethanol. Why? To dissolve the fat if it's present. After that, take this test tube and pour the content in cold water. If we don't have fat, so the solution will be clear, nothing there. But if we have fat, so it will be turbid or milky or cloudy. Why? Because the fat under low temperature will be solid and white. The last one, vitamin C. Vitamin C. What is the test? Testing for or vitamin C test. The reagent DCPIP. DCPIP is blue in color. So if we have the food solution, we'll add few drops of the reagent DCPIP, and the change of the color from blue to colorless will be according to the concentration of the number of the drops which use according to the concentration of vitamin C. So if we have few drops, it means strong vitamin C solution. If we have many drops, so weak vitamin C solution. So according to the number of drops, and to change the color of the reagent from blue to the colorless, the concentration of vitamin C will be there.